Hello, everyone. Well, thanks everyone for inviting me to this greatly amazing conference in Evolution 2025. Actually, it's quite nice because it's something that I have always wanted to attend to. I mean, I'm quite of a fan. And then, well, so let's start. Well, it's my presentation. It's called, let me move a little bit this, so it's smaller. Nice. No, so good. So it's called Genome Evolution and Sexual Selection Among Mammals. Well, as you can see here, I have well, I'm in University of Bath and the Minno Center for Evolution. Here you can see a little bit of the building. It's quite a pretty place, but also this project's been co-hosted by UNAM University in Mexico. It's quite of a joint venture for this project. So, okay, great. So here, what the main thing behind this presentation is to present to you one of our last papers. It's called Sexual Size Dimorphism Among Mammals. It's how it's linked to gene family size, therefore genome evolution, right? So that was we published last year-ish. And then, well, let's start here. It's sexual size dimorphism. Well, a lot of us, we know that sexual size dimorphism, it's a proxy for sexual selection, right? So that's, and in our lab, we are quite interested about the implications or the impact of sexual selection on genome evolution and also on phenotype evolution, right? So that's why here we took 124 mammal genomes from different sources. Well, no, pretty much mainly from NCBI, from RevSec. And then we mapped the orthologies of those between those mammals. And then we correlated that with genes, sexual size dimorphism, right? And then we thought, okay, let's see what happens, right? As many times in a lab, you have like a hunch and some decent background about what is going on, and then you just jump into the abysm, right? And interestingly, what we found it's that brain functions and brain development, it's been linked to sexual size dimorphism, but maybe you will, but not in the way that you might be thinking at this moment, or maybe you do. And also it's also linked with olfactory receptors. And then let's go and see what happens. Interestingly, what we found is that gene family expansion, we found several gene families, such around 340 more or less, and then associated with SSD, with sexual size dimorphism. And these gene families, these have several genes inside of them. We run a Go enrichment analysis, and then we found, as you can see in the top, in the lower corner in the right side here is sensory perception and smell. The small part, it's actually the expanding gene families associated with with sexual size dimorphism, which are mostly linked to sensory perception of a smell, which in some kind of way could have like some kind of a association with sexual selection. But the most interesting part of this finding is that the contracted ones, the contracting gene families in quotation marks, are actually quite highly associated with neuron migration, as you can see, also neuron differentiation, etc. So the interesting part, or the key point here, is that these are the contracting gene families. What does this mean, right? So that will mean that a species that does not present a strong sexual size dimorphism are actually investing in brain development rather than other functions, such in this case, olfactory sensory, right? Okay, then, as we said before, so expanding genes are linked to olfactory receptors, which in some kind of way could be related to mate choice and other survival functions more linked to natural selection, perhaps. We didn't really test that. But then brain development genes are linked to the species that have less sexual dimorphism, which is quite cool. And in some kind of way, you think about it, has some sense, right? Taking in account that maybe sexual selection, it's among mammals, there is a lot of competition. It's a lot of male competition. So if the species starts investing in like the social brain hypothesis, right? So by interaction, creating some kind of a society, the brain starts to develop, and so they have to start to have more complex behaviors, such as parental care or dealing with other peers of their same species, right? Then, okay, so another thing that we did is a differential gene expression analysis of these genes that we found correlated. What we found, interestingly, is that the contracted genes, the ones that are linked to brain development, are highly expressed in early stage of development, such as you can see in the green square. So at the beginning, before birth, it's highly expressed in these subjects, and by the pass of the time, they start to decrease the expression of those genes. So in some kind of way, what we can see is that, okay, at the beginning of this differential gene expression in humans, at the beginning, it's brain development. It's a highly expressed pathway and such, right? And then, but the contracted ones, the contracted genes, the expanding genes are the other way around. At the beginning of the developmental stage, it's not as highly expressed, but then by the pass of the time, it starts increasing the expression. So through adulthood. So we also did a fold change analysis with trying to compare differential gene expression among males and females, and also between fetal and developmental stages, but we couldn't really find like a concrete answer for that. So we could not really find a sexual differentiation among the genes that we found so far. So as you can see here, right? Then what we can see is that in this project, we saw that, okay, that we use sexual size dimorphism, which is a proxy for sexual selection, and apparently it has an impact on gene family size. What is this telling us in some kind of way? That apparently sexual selection has an interesting story behind how our genomes and also how different organs, such as the brain, have evolved among mammals, at least, right? So what's next, right? So we analyze here sexual size dimorphism, but in the future, yeah, that's what I was saying, right? That in this case, males via SSD, it's linked to sexual selection, and then brain size, it's more like a social behavior, social evolution, etc., which starts more conversation than giving answers. And then the next step will be in our lab is to test size against gene family size, that we haven't really seen much about that. And also compare both of our analysis. And this will make like a more well-rounded story around sexual selection. And we are working on that. And the results looks quite promising so far. And then let's start a summary here. So our key findings is that gene family size changes, evolution, it's associated to SSD. And therefore, probably some kind of connection to sexual selection, because this is an associative analysis. So we cannot say that we cannot infer causality, but yeah, right? So these implications, well, as we said, sexual selection, genome evolution, they have some kind of potential link. It's a very interesting story. And in the future, as we said, we are gonna expand our analysis to test size, but also we would like to make the connection between longevity, also brain development, and perhaps some potential linkage with sexual selection in this case, which also actually we are writing some stuff about that. And let's hope that we can actually publish that paper soon so everyone can see it, right? And then let me show you some of our team here. It's thanks to these people that they are very important people that without them this probably would not have been possible. With Professor Matt Wills, Thomas Kelly, very good mentors from the Milner Center for Evolution, and also a really big mentor, Dr. Araxi Rutia, that she used to be with us in Milner Center, but now she's in UNAM University in Mexico. And yeah, but this makes a very nice team. We have known each other for a lot of time. But yeah, thank you very much. And I don't know if you have any questions.
Thank you very much. Clap, clap, clap. Questions for Benjamin here. I guess you can put them in the chat, or you can raise a hand, and I can unmute you either way. I guess I have a few. So yeah, so you might have said it and I missed it, but you were talking about differences in gene expression in the brain. Was that just the brain writ large, or was it specific parts to the brain, like certain types of brain structures, or yeah?
Oh yeah, actually for the brain expression part, we actually use neocortex and other parts of the brain. And we could find, our analysis said that most of our genes, I think so, they were expressed in the frontal cortex. But also overall, it was highly expressed in the whole brain.
Yeah. So I guess the link with these brain development genes, it's sort of like I don't know if you hinted at, or maybe it was me like inferring it. But so I guess, do you think that you actually have a trade-off between sexual size dimorphism and brain development? So yeah.
Sort of. That's what that's what the data in some kind of way infers, that apparently because in this case, in this correlation, what it's telling us is that the species that have strong or higher sexual size dimorphism those gene families are linked to olfactory senses, olfactory receptors, and other functions. Meanwhile, the ones that were, let's say, in the other side of the graph, or in the other side of the correlation, which are the species that have lower sexual size dimorphism, are actually going to the path of brain development.
Yeah, so I guess I'm just curious whether or not that, like, if you see gene expression, you can sort of infer, okay, this is higher investment in the brain versus lower investment in the brain. And I guess part of my I guess we're getting close to time here. Zoe cut me off at 14:30. It's because like, I thought a big thing about the brain development is actually cutting certain neural pathways, right? So like babies have lots of connections in their brain, and then they trim those as they develop. So whether or not there's a gene expression or pattern for the genes themselves that infer greater investment in brains.
So Benjamin, if you want to respond to Becky, but please stop sharing your slides, and Ken, we're gonna have you start sharing.
Sure. Okay, sorry for that. Well, yeah. Yeah. Oh, good. Yeah, right.
So then, Ken, if you can go ahead and start sharing. Thank you.
Yeah, well,
I got carried away in my job as moderator.
In this case, the thing is that we found that gene expression in the brain for all the genes that we found. So in some kind of it's tricky, right? In order to find that. But thank you for that.
Thanks. Our next speaker is Ken Toyama. He's from the Universidad Nacional Mayor de San Marcos. The title of the talk is The Allometry of Bite Force Dimorphism in Anolis Lizards, a reappraisal of Rensch's rule. Take it away, Ken.
Thank you. Hi, everybody. I hope you can see the slides well. And today I will tell you a little detective story. And the story starts with this, that there is a great diversity of sexual dimorphisms in nature. And as was said in the previous talk, the most famous is probably sexual size dimorphism, or the differences in size between males and females. So if we start to zoom out and go to the macroevolutionary level, we will start seeing some patterns. The most famous of these patterns is probably Rensch's rule, which is an empirical pattern where the ratio of male size to female size is larger, the larger the body size of the species. So in this plot, if each dot is a species and we have these three clades, we will see that the degree of male bias, sexual size dimorphism increases with species size. So Rensch's rule studies have become quite popular in the last few decades. And since its postulation, it has been very influential for many important works, like the studies on sexual selection on primates by Clayton Brock, the genetic models for sexual dimorphism of Landy, and this also famous meta-analysis of the allometry for sexual size dimorphism by Daphne Fairbairn. But who was Rensch? Bernhard Rensch was a German biologist. He was interested in many different topics, but especially in macroevolution, biogeography, biophilosophy, and he was an important contributor to the modern evolutionary synthesis. So regarding Rensch's rule, Rensch presented his rule in two of his works, Rensch 1950, which is a paper, and Rensch 1959, which is probably his most influential work, which is the book Evolution Above the Species Level. And this takes us, well, actually, virtually all studies that have something to do with Rensch's rule will cite either Rensch 1950 or Rensch 1959 or both. And during my PhD, I became quite interested in collecting classic books in ecology and evolution. And at the same time, as a PhD student, I was working a lot with Rensch's rule. So it just seemed appropriate for me to get a copy of Evolution Above the Species Level, which takes us to the next chapter in the story, which is Ken gets Evolution Above the Species Level. So I ordered the book, of course, and I got it in the mail. I was very happy. And the first thing I did was, of course, to go to the lines in which Rensch writes about his rule. And he writes very little. Basically, he says that in numerous animal groups, sexual dimorphism in secondary sex characters increases with body size. And I'm emphasizing here secondary sex characters because he didn't emphasize, or he didn't mention explicitly sexual size dimorphism. In fact, he was using as examples the relative differences in individual body parts, like the antennae of some beetles, for example. So he showed how this relative difference was higher in larger species than in smaller ones. When finishing the lines that he wrote in this book about his rule, he said that in mammals, the anatomical structures used as weapons, such as antlers, horns, teeth, and the like, provide more examples of this rule. You just need to look around. So for me, this was quite a surprise, because I didn't find an explicit mention of a relationship between sexual size dimorphism and species size, which is supposed to be the definition of Rensch's rule. But of course, I still had to revisit another source, which was Rensch 1950, which takes us to the next chapter of the story, which is Ken reads this paper from 1950. Of course, I had a problem because the paper was written in German. So I used the help of Google Translator and my ex-lab mate Alex Tinius, who is German, to help me translate this paper. So the title will be translated as something like The Dependence of Relative Sexual Differences on Body Size. So in this paper, Rensch first writes about different proportioning rules he observes in nature. For example, among related species, the larger ones tend to have smaller or relatively smaller brains, relatively smaller hearts or eyes. So he describes more of these rules, and eventually he shows, or he wants to show the world a new rule that he thinks he has discovered, which says that in species of the same family group, the relative sexual difference is more substantial in the larger of these species than in the smaller ones. And again, I'm highlighting the relative sexual difference because again, he's not specifying sexual size dimorphism as the definition of Rensch's rule that we know of today. Yeah, so, but then he starts exemplifying the rule and he mentions, let's first compare the most common sexual difference, the body size of the two sexes. So finally, Rensch says something about sexual size dimorphism. And he compares and shows how among related species, sexual size dimorphism or male bias sexual size dimorphism is higher in the larger species. But eventually Rensch moves from body size and starts showing examples that involve other individual body parts. And again, we see this figure like in the book. Rensch starts talking a lot about beetles and shows how the relative sexual differences, some traits like the antennae or the mandibles, are higher in larger species than in smaller ones. So yeah, and in fact, Rensch concludes in this paper that different animal groups show a relatively greater sexual difference in larger species with regard to morphological sexual characteristics and body size. This was very interesting because by using the term sexual dimorphism, Rensch did not discriminate between sexual size dimorphism and other types of dimorphisms in this paper or in the book. So what we know as the definition of Rensch's rule maybe shouldn't be specifying sexual size dimorphism, but any relative sexual difference. So let's test this rediscovered knowledge in Anolis. So with the help of my collaborators, Jonathan Lossos, Anthony Herrer, and Luke Mahler, who was my PhD supervisor, we tested Rensch's rule in body size, but also in another trait, in this case, bite force as a measure of performance. So we considered 71 Anolis species and a measure of size, which will be snout bend length, and also, of course, bite force. So what did we find for Rensch's rule in body size? So we found this pattern. So as you can see, we divided Anolis in two categories, in island and mainland species. And this is because island and mainland environments are different ecologically. In theory, stronger sexual selection should occur in islands, but still, we found that both faunas follow the rule, follow Rensch's rule in body size, which means that higher male bias sexual size dimorphism evolves in larger species. So what about the Rensch's rule in Anolis performance, in bite force? So for this, since we are working with relative dimorphisms, we first had to size correct performance, which is a bit tricky, but we just had to consider this allometric exponent of bite force, which we estimated empirically with our data. Then to sort of revisit our expectations, to find Rensch's rule in bite force means to find a positive relationship between size corrected performance dimorphism, which here is our measure of relative sexual difference, and body size. And this will occur if the evolutionary allometry of males has a higher evolutionary slope than the allometry of females. But then what if we find no Rensch's rule? We propose here two different scenarios. As you can see, there is no relationship between size-corrected dimorphism and size. So in the first scenario, males and females share the same evolutionary allometry. And in the second one, males and females differ in the allometric intercept, but not in the slope. So what we found for bite force was this. If you remember, we had these three scenarios, these two no Rensch's rule, this one here Rensch's rule. And what we found is that bite force in Anolis does not follow Rensch's rule. Basically, mainland species follow this scenario here, and island species follow that scenario there. So we found no Rensch's rule in Anolis or in any of the categories, but we found higher relative bite force dimorphism in island species, as you can see in this scenario. And we also found out that it was due to relatively larger heads in males relative to females. But this difference was not dependent on size. So we actually tested Rensch's rule in relative head size dimorphism as well, and we found also no relationship. So what does it all mean here? So this is what we found for Anolis. We found that Anolis follow Rensch's rule in body size, which means that male bias sexual size dimorphism is higher in larger species. But we didn't find Rensch's rule in bite force or in head size, which means for the case of head size, that the relative difference in head size between sexes is constant across the size distribution. So then what would happen if we look at other similar systems? Are we going to find the same patterns or not? Of course, this means that performance dimorphism is mainly determined by size differences in this case, but there might be other systems in which we find different things. For example, in the group of Timon and Podarcis lizards, we see that there is Rensch's rule followed by body size, which means that sexual size dimorphism, male bias sexual size dimorphism is higher in larger species. But we also see that head size follows Rensch's rule. This means that the relative difference in head size is higher in larger species, even if there is no size involved. This means that in this case, performance dimorphism is going to be determined by size differences between sexes, but also independently by dimorphism in a secondary sexual character like head size in the case of lizards. So why do Timon and Podarcis follow different patterns than Anolis? Why do similar systems show different patterns? We don't know. Somebody will have to study this in the future. So the modern Rensch's rule is not followed by all animals, like in the previous example. For example, we have here a group of harvestmen and a group of weevils, and we see that there is an association between male bias sexual size dimorphism and body size. But now we know that Rensch's rule was originally a pattern applicable to more than just size. So if we could try to test Rensch's rule in a secondary sexual character. In harvestmen, we could test it in the length of the legs, and in weevils, we could test it in the width of the mandibles. And in fact, you will see that Rensch's rule is true in these cases. So in both cases, this means that performance dimorphism will be higher in larger species, not because sizes are different between male and female, because we saw that that dimorphism is constant across sizes, but because there is an allometry of shape dimorphism. So here we can start asking new questions, like why sexual size dimorphism allometry doesn't evolve in some systems? Is there some sort of constraint in body size such that they have to rely on the evolution of positive allometry in secondary sexual characters? Again, more studies will have to look at this in the future. And just to finish some short conclusions. First is that what is known today as Rensch's rule is just a special case of Rensch's original rule of sexual differences. And that, of course, allows us to propose new questions and new hypotheses now that we can test Rensch's rule in other traits. Then we saw that dimorphism in body size and individual body parts can show different patterns of allometry, as we saw just now. And finally, that an overwhelming majority of Rensch's rule studies has focused on the relationship between size dimorphism and body size. Basically, 99% of these studies have studied that relationship. So who knows what else might be out there when we look at the allometry of dimorphism in other traits, right? So yeah, this short talk was based on these two papers. I invite you to give them a read if you're interested. They were just published this year. And yeah, that's all I have for today. Thank you. You can write me to that email. You can ask me any questions you want. So thanks again for your attention.
Great. Thank you very much. There are a couple, or there's a question in the chat. You might have answered it, but I'll go ahead and. Sorry, my dog's bark. The question says, I'm interested in knowing if this is similar in invertebrates where extreme sexual size dimorphism is quite normal, just a thought. And then there's a follow-up to that. Yeah.
Oh yeah, so somebody was, no, that was for Ben, I guess.
Oh, sorry. I guess I'm sorry, I messed up. Okay, well, are there other questions for Ken? I have one, but yeah. But I think I see Benjamin raise his hand. Benjamin, did you have a question?
Well, yeah. Well, I mean, in my case, it was more like okay. So you analyzed bite force, right, and be correlated with Rensch rule, right?
Yeah.
Yeah, but the thing here is like, I mean, what would be the implications of bite force with sexual selection? And maybe it's a very, I don't know how you can explain that in a biological context, like more or less.
Sorry about that. I guess I should have explained that, like lizards fight, male lizards fight each other, biting each other. So bite force is a very important measure of performance for lizards. And of course, larger lizards will bite harder, but even if they just keep the same shape, right? But We have the cases in which head size can become disproportionately larger, as it happened in some lizards, but not in anoles, as we found. But yeah, basically, is that bite force is important for sexual selection in this case.
For lizards mainly, right?
Yeah, and probably for other animals as well.
Great.
Like
Thank
beetles.
you guys very much. We're out of time here on that one. Thanks again. Our next speaker is Aviad Heifetz from the Open University of Israel. The title is What About Male Quality? A Model to Predict Male Investment in a Single Female. Take it away.
Okay, can you see my full screen? Okay, very good, thanks. So this is joint work with my colleagues, Yael Lubin and Michal Segoli from Ben-Gurion University and Jutta Schneider from University of Hamburg. They are studying spiders and other species, and I'm a game theorist, I'm a mathematician, so we joined forces here together to ask a question which is of joint interest. So we know that in classical sexual selection theory, it is the males who are who are those who are supposed to chase as many females as they can, while females should be choosy, okay, because they are limited by the number of offspring that they can raise. But there are also examples of to the other side. Okay, so and this is the case. This would be the case of monogeny. It's more rare. It's less well understood. This is the case where males invest exclusively in one female, while the females can mate more than once or multiply. And this is known in spiders, especially when there is extreme sexual size dimorphism, where females are much larger than males. And the way males try to get to monopolize females is by long courtships, long copulation, guarding the female after copulation, leaving some part of the genitals as plugs in the genitals of the females. And the most spectacular example is where males actually sacrifice themselves in order to get monopoly. So they expose their body so that the female can eat them, just to prolong a little bit more the copulation, and in this way maybe leave some of the genitals as plugs, for example. What could be an explanation for that in spiders? So one important factor in some spider species is that males mature fast, while females take more rounds until they become sexually mature, and they become much larger by then. So in large parts of the breeding season, there would be many more males than females. So much more competition among males over few females. And also females build their own nests, and males have to travel to the female's nest. And if they want to move to another female, they have to make the way between them, and they're exposed to predation. So what do we know from existing game theoretic models? So the main messages are that the harsher the condition is for males, that is when they are many more males competing over one female, and there is a lower survival probability when traveling across females' nests, then it is more likely to see monogeny. That males, once they reach a female, they would try to monopolize her and not try to get to another female. And what we do in this theoretical paper, we want to look at one additional dimension, which was not addressed in the previous theoretical papers, and that's male quality heterogeneity. Okay, because after all, when females are so large, they can be choosy. Choosy means being able to accept some males and not to accept others. So that's what we assume in the model, that the female indeed can assume the male quality by appreciating the male size, by appreciating how agile he is on her nest when he approaches her, and she can repel males that she dislikes. Okay, nevertheless, it might not be wise for her to repel all males, okay, until the most preferred highest quality male arrives, because after all, there is a limited period in which she can mate. Okay, so if she rejects males for too long, okay, then at the last minute, she would have to accept a random quality male that approaches her. So there is a trade-off here. On the other hand, if she allows the males to mate with her, so first of all, he might be able to monopolize her, and if not, then there would and another male makes it to her, then there would be sperm competition between the sperms of the two males. So what comes up from solving this problem from the perspective of the females is to do the following. At the beginning of the breeding season, set a threshold, C, okay, and then accept males only with quality higher than that. Towards the end of the breeding period, accept only males with higher quality, okay, because they would have higher quality sperm. And in particular, if the female did not accept anybody, she repelled the male in the first breeding opportunity, then just accept anybody who arrives at the last moment. What about male? They should decide whether to try to monopolize the female, maybe at the cost of their life, or try to copulate with the female that accepts him and try to forgo monopoly and try to mate with additional females. So what we know from the existing models, we get the same message here. Okay, if conditions are harsh for males, so many predators, many competing males, then yes, it's better to try to monopolize the female and opt for monogamy. The somewhat surprising result, okay, is that the best of best males would actually do try to look for other females. And why? Because there's no point of foregoing additional mating opportunities. I mean, if I'm a superb male, then additional males who might approach the same female would probably be of lower quality than myself, and she would reject them. And so there's no point of giving up the opportunity to look for additional females. So we're looking for the ESS, for the evolutionarily stable strategy, where the the strategies of males and females are characterized by these thresholds, okay? What is the minimum quality C of males that females accept early in their fertility period? And what is the threshold quality for males such that if they are of higher quality than that, then they would look for additional females? So I'm not going to show you any formulas due to the shortage of time. I'm just going to show you how the results look like for some reasonable assumptions on parameters. So male quality is you know, we assumed it is distributed between 0 and 1. And with you know, with more males with medium quality than very low or very high. And what we are showing here next to male quality is also what are the conditions for males. Okay, when males, this is the situation where competition is very so conditions here are good for males. Okay, sorry. So male survival is relatively high. Sex ratio is maybe also close to 1. So not too many males to females. Here conditions are very harsh for males. Okay, either low survival probability if they move to the next female, or many males to each female. And what we show here is the ratio between the monogeny strategy to bigeny strategy. Okay, so for trying to look for additional females. What we see in the result here is that there is this small area, okay, where only here monogeny is better for the males. Okay, so when conditions are harsh for males, okay, some of the males, okay, those with lower quality, okay, would want to stay with the female that has already accepted them. Other ones, okay, would want to, better quality males would try to look for additional females. The transparent blue square here just shows where the ratio of the two strategies is 1. Let me just show you this on here. Maybe you can see a bit better. So here we see the same, the conditions for males. Here conditions are good. So I forgot to mention in the parameters I'm showing you, females accept males only if they are above 0.47 something, okay, between 0 and 1. And here we see in this region, okay, that the better quality males, okay, look for additional females. The lower quality males, okay, try to stay with the same female.
We're gonna need to finish in like the next 30 seconds or so.
I'm done.
Oh, excellent.
Thank you.
Sorry. Thank you very much. Yay. Questions here? Benjamin, I see your hand up again.
Yeah.
Hi. So, can you recall which parameters you use for male quality? I think you use like a sperm quality and others, right? If I'm not mistaken.
So the assumption is that the female can assess the quality of the male. Okay, they cannot assess directly the sperm quality, but just how big the male is, how agile it is on the female's nest.
Yeah, okay, like it's in an overall, like it's based on the female's perception, right?
Exactly. Yes.
Okay, cool. And then can you go back to your graph in 3D?
I'm
Well, Zoe does
yeah,
not allow me
sorry, we're
anymore.
not going to have time. We need to get on to the next
Don't worry
one.
about that. So
Okay.
the thing here,
Yeah. So Nicole, do you want to go ahead and share your screen?